Okay, so now we're gonna be talking about microscopy. Okay, um, this is microscopy. Ooh. So there's something called the ranking biological scale. That means you can actually order things through the relative size of the structures. So let's consider what is big, what is large. So think of a sailboat that's very large, but let's think of a mitochondria that's very small. And viruses are even smaller, and an atom is like near the smallest. So yeah. Let's talk about some light microscopes quickly. So light. So these include stereo microscopes and compound microscopes. So stereo and then compound. So let's talk about these two. So stereo microscopes actually allow you to observe the surface of the sample. So some of the pros are that you can view the living sample, but there are a few cons to this. So it has very low light resolution and compared to specifically the compound. And so let's talk about the compound now. So you can see the thin section of a sample here. And some of the pros to this is that you can see the single cell layer of a living sample. But con would be it requires staining for visibility. So something just key to remember is it requires staining. Okay, there's no more light ones. Let's also talk about phase contrast microscopes. Phase contrast microscopes. So for phase contrast microscopes, you can see the detailed observation of the internal structures. And some of the pros with that is that it has good resolution and contrast, but some of the cons is that it's not good for thick samples specifically. So if it's thick, look, thick, it's not good. Not good for phase contrast. And it can also have this halo effect around the sample perimeter. Let's continue. We can do the scanning. Oh no, we can do the confocal laser scanning. Scanning plus fluorescence. Oh, oops. Okay, so it has very thin slices. So the sample is actually left intact, and it's often used for chromosomes. So some of the pros are you can observe specific parts through fluorescence tagging. But some of the cons is that it can cause artifacts. And of course, it can be used without fluorescence as well. So we can do also the scanning electron microscope. So the SEM, SEM. So some of the pros with this one is that you view the surface of a 3D object in high resolution. But the con with this would be that you can't use it on living samples and it requires extensive prep, so drying and coating. Then we also have the Cairo version of this, so the ChirosEM, which in this case, some of the pros is that the sample is actually not dehydrated. So Cairo is actually used to make observe a sample in a more natural form. Along with that, some of the cons include that, again, it cannot be a living sample and it must be frozen, so it can cause artifacts. Finally, we can also go on to the transmission electron, so microscope. So the TEM this time. So this one, you can actually see very thin cross-sections in high detail, along with the internal structures in extremely high resolution. But some of the cons with this is, again, no living samples can be observed, and the prep is actually quite extensive and very expensive. So yeah, let's also talk about the electron tomography. So with the electron tomography, it builds a 3D model alongside using the TEM, and it's not a microscope technically. This electron tomography. So again, the pros are you can see the 3D model of whatever you are observing, and you can see relative sizes. But the con is again the same as TEM. It can't be used for living samples, and the prep is very extensive and expensive. So let's continue on to centrifugation. So with centrifugation, which I definitely spelled wrong here, so let's just correct that. Centrifugation. And basically, there's a device called a centrifuge which spins and separates a liquefied cell into layers based on its density. Remember, this must be homogenized before you centrifuge it. So there's two types. There's differential or density centrifugation. So you can do the density one, which is just based on the most dense go to the bottom, the least dense go to the top, or there's differential, which is based on density, shape, and speed. So the differential actually creates continuous layers of sediment, and it is the insoluble compounds are found in the pellet, and the soluble is in the supernatant. The order of separation goes along the lines of it starts with the nucleus, and then it goes to the mitochondria, then the chloroplast and lysosomes, then the microsomes and small vesicles, and then finally ribosomes, viruses, and other larger macromolecules. So biggest to smallest, basically. Okay, so that was microscopy, and we will continue next time with PCR, I think.